chromosome structure. Generally, eukaryotic cells containing chromosomes in the nucleus. Eukaryotic cells containing chromosomes in the nucleus. So first, the chromosomes were first discovered by Walter Fleming in 1882. In the nucleus of the cell, the DNA molecules is packaged into thread-like structure called chromosome. DNA molecule is packaged into thread-like structure called chromosomes. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. Mean the chromosome structure supported by formation of histone proteins only. The chromosome's unique structure has a few key parts. Each chromosome has two short arms called P arms and two long arms called Q arms. These arms are held together at the center by the centromere. Mean the centromere is the center part of chromosome. The tip of chromosome are capped by section of DNA called telomere, which carries, means the chromosome carries genetic information from one generation to another generations, and the chromosomes play vital role in the cell division, heredity, variation, mutation, repair, and regeneration. These are the functions of the genetic material like chromosomes. Now we discuss about the clear structure about the chromosome. See, it is the chromosome overview. Mainly the chromosome containing two major parts. One is centromere and arms. Majority of the chromosome containing arms, the two arms are not in equal condition, not in equal structure. Sometimes one arm is in large in size, one arm is short size. Short size of arms are called P arms. Large size of arms are called Q arms. They are connected with the center part, a spheroidal globe-like structure called centromere. Around the centromere or from the centromere, chromatin thread-like structures are formed. The chromatin thread-like structures are also called another form of DNA. Chromatin thread-like structures are also called another form of DNA. They are also called chromatonemata. Chromatonemata are chromatophores or DNA. They are carries genetic information. The chromatonemata compactly arranged by the histone proteins in the arms. So during the formation of arms, the chromatonemata covered with colorless liquid. Around that chromatonemata, colorless liquid is present, is called matrix. Matrix is the protective layer of the chromatonemata. Then outside of these matrix, the surface of matrix is called pellicle. During the formation of arms, the chromatonemata shows two colors. The two colors are called heterochromatin, euchromatin. Euchromatin and heterochromatins are present in the chromatonemata. Euchromatin is a light color. When we treat with acetocaramine solution, the chromatin thread-like structure always in light color. That areas of chromatonemata are called euchromatin. They are the true genetic materials. They are involved in the hereditary characters and transcription characters and other characters also there. Euchromatin is important for genetic material. When we added the acetochromatin to the chromatomata, some chromatin thread-like structures are dark in color. The dark color chromatin thread-like structures are called heterochromatin. They cannot participate in heredity. They are sleeping genetic material. So they cannot involve in the hereditary characters. During the formation of these chromatomata, they shows euchromatin and heterochromatin. Mainly around the centromere. Around the centromere, chromatomata are make a primary constrictions. Mean the chromatomata start from the centromere only. So they are started from the centromere. The started chromatomata are also called primary constrictions. In this primary constriction, euchromatin genetic material is in more number. So in primary constriction, euchromatin is in high concentration and heterochromatin is completely absent in the primary constriction. During the formation of arms and far from the centromere, the chromatomata shows euchromatin and heterochromatin, and they are also shows in the arms-like structure secondary constrictions. The secondary constriction look like a notches-like structure in the formation of arms. The notches-like structures are called secondary constrictions. In this secondary constriction, heterochromatin is in more percentage and euchromatin is in less percentage. But in primary constriction, euchromatin is 100%. Mean the loosely arranged chromatin fibers. Loosely arranged chromatin fibers are called primary constrictions. Tightly arranged chromatin fibers are called secondary constrictions. At the end of these two arms, terminal parts are easily identified. The terminal part of arms are called telomeres. The telomeres mainly helps in identification of terminal part of chromatin fibers. Chromatin thread-like structures or end of the DNA. So we identify telomeres. It is end of the chromosome part or it end of the DNA material. And at end of these two arms, spheroidal structures are present. The spheroidal structures are also called satellites. Mainly the satellites helps in avoiding of mixing of genetic material between two chromosomes. The satellite mainly avoid of mixing of genetic material, changing of genetic material between two chromosomes. And around that centromere, kinetochores are present. The kinetochores are always connected with the spindle fibers. The spindle fibers are connected with kinetochores, and during the division of chromosomes, they are helps in the division of chromosomes with the help of spindle fibers, and chromosomes cleaves into two half parts during the mitosis and during the meiosis. We clearly observed the formation of euchromatin and heterochromatin in the chromatin thread-like structure. The major differences are here is euchromatin and heterochromatin differentiation. Here it is euchromatin participated in transcriptionally active, means it involves in the transcription mechanism, and heterochromatin cannot participate in the transcription. So heterochromatin DNA transcriptionally inactive and euchromatin DNA is loosely packed. Heterochromatin DNA is tightly packed or highly packed. And euchromatin actively present in prokaryote and eukaryotic genome. Eukaryotic genome and prokaryotic genome containing active euchromatin, and heterochromatin present in only eukaryotic genomes. Prokaryotic genomes didn't have any heterochromatin in their genome. And in euchromatin, genetically active. Genetically active means they're genetically involved in hereditary characters. They can pass genetical character from one generation to another generation by the euchromatin genetic material. But heterochromatin genetically inactive. It cannot involve in the genetical hereditary characters. And euchromatin present in inside of the nucleus, present at the center region of the nucleus. Center region of the nucleus, chromatin thread-like structures loosely arranged, they called euchromatin. But heterochromatins are present at the peripheral of the nucleus. Peripheral means not present at the center region. They present at the peripheral region of the nucleus. And euchromatin stain in acetocarmine, stained with acetocarmine solution, it shows light color only. But heterochromatin, it shows dark color with staining of acetocarmine chemical. And early replicative means it ready to participate in the replication. And heterochromatin late replicative, it is in sleeping mode. It cannot participate in actively replication process. Then they are not in sticky condition of euchromatin. And heterochromatins are usually sticky and mean compactly arranged their structure. They are closely arranged, compactly arranged their structures called usually sticky structure. And euchromatin allow gene to form a protein, to make protein by euchromatin only. But heterochromatin regulates genetic integrity and it controls gene expression. The gene expression and gene integrity or genetic integrity controlled by the heterochromatin eukaryotic living organisms. And low genetic density by euchromatin, high genetic density by heterochromatin. Genetic density means compactment or arrangement of genetic materials. Genetic materials compactly arranged is called high genetic density, mean present of genes on the one location. But in euchromatin, the genes are loosely arranged. So they are loosely arranged, they containing only low genetic density. And the euchromatin in chromosome, it consists of 2 to 3% of genome only. But in heterochromatin, it consists of 97 to 98% of part of genome. So it is in large quantity, but these are the cannot involved in the heredital character, cannot involved in the formation of proteins. Mainly the heredital characters, formation of proteins done with only euchromatin genome. They are the active genomes in the chromosome. It is a chromosome structure in eukaryotic cells.